Imagine your immune system as like a highly organized, heavily armed military.
Right, a massive biological army.
Exactly. You've got your general strategizing in the back, your scouts out there gathering intelligence, and your standard infantry just marching in line.
And in the T-cell world, which is essentially your specialized infantry, almost every single soldier wears a very clear, highly visible uniform.
Yeah, biologically speaking, that uniform is a protein marker sitting right on their surface. Usually it's either a CD4 badge or a CD8 badge.
Right. You look at them under a microscope and you know exactly who they are, you know what their orders are.
And it's a beautifully binary system that scientists and doctors have, relied on for decades.
It really is. I mean, those CD4s, they're your helper T cells. They act like the commanders coordinating the attack from the rear.
While the CD8s are your cytotoxic cells, right?
Exactly. The CD8s are the infantry actually pulling the trigger on the front lines. It's neat, it's easily categorizable, and quite frankly, having that level of predictability in biology is incredibly comforting.
But today, in this deep dive, we are stepping far, far away from those standard ranks. Today, we are looking at the rebels.
The rogue operatives.
Yes, the operatives who flat out refuse to wear either uniform. We are talking about double negative T cells, or DNT cells for short.
Right, because they lack the CD4 badge and they lack the CD8 badge. They are double negative.
And for a long time, immunology just kind of didn't know what to do with them.
No, they really didn't. They've historically been treated as this weird afterthought, just, an anomaly that didn't fit neatly into our textbooks.
But a massive, absolutely groundbreaking new study has just completely rewritten our understanding of these cells.
Yeah, we are talking about a pan-cancer single-cell RNA sequencing atlas.
Okay, let's unpack this because the scope of the sources we have today is truly staggering. They didn't just look at a handful of samples in a Petri dish.
Not at all. They mapped over 150,000 of these incredibly rare, rule-breaking DNT cells.
Right, and they looked across 23 entirely different types of cancer. They pulled from, I think, over 2,000 distinct tissue samples.
It's a monumental data set.
So our mission today is to explore what these renegade cells actually do, how tumors actively try to brainwash them, and why understanding their secret lives might be like the ultimate key to unlocking better cancer immunotherapies.
And to really grasp the gravity of this atlas, we first have to understand the foundational biology of why they were just ignored for so long.
Which comes down to numbers, right?
Exactly. DNT cells make up a tiny, tiny fraction of the T cells circulating in your blood, usually just about 1 to 5%.
Right, which makes them incredibly rare. I like to think of regular T cells as a standard deck of cards for you listening. Your CD4s are the hearts, your CD8s are the spades. Very standard, very expected.
That makes these DNT cells the jokers of the deck.
Yes, absolute wild cards. But, I have to push back on the premise here. If they only make up 1 to 5% of our T cells, why not just focus on the other 95%?
It's a fair question.
I mean, aren't they mathematically just too rare to make a meaningful difference against a massive tumor?
Well, it's a completely logical assumption, but what's fascinating is how it reveals a massive blind spot in our own scientific history.
So?
Historically, the easiest way to study the human immune system was to draw blood.
Right.
It's accessible, it's minimally invasive.
Yeah, you just roll up your sleeve.
Exactly. But blood is actually a terrible representation of what is happening inside a solid tumor. Think about it. Blood is just the transportation network. It's the highway.
Oh, I see.
Studying blood to understand a tumor is like trying to understand a brutal trench war by interviewing soldiers on a train hundreds of miles away from the front lines.
Ah, so it's the classic looking for your keys under the streetlight just because the light is better there problem. We study the blood because we could easily get to the blood.
Precisely. We were looking in the wrong place, so when the researchers built this massive pan-cancer atlas, they intentionally didn't just look at blood.
Right. They took biopsies of the actual tumor tissues, the adjacent healthy tissues, and normal organs.
Yeah. And because these cells don't have the standard CD4 or CD8 markers, scientists have to classify them by looking at their primary sensor, their T cell receptor.
And those sensors divide the DNT cells into 2 main family trees, right? The alpha-beta lineage and the gamma-delta lineage.
That's right. You've got the alpha-beta, or UC, and the gamma-delta, or GA.
Mm-hmm.
And when they finally looked at the actual tissue, they found that the gamma-delta lineage cells are what we call tissue resident.
Meaning they don't commute.
Exactly. They don't just circulate in the blood waiting for an emergency call. They permanently live directly in the organs.
Like the lungs, the skin, the intestines?
Yep. They are stationed right where the battles actually happen.
Wow. Okay, that dramatically shifts the perspective.
Yeah.
So let's do a quick recap for you listening before we dive deeper into their actual day jobs.
Good idea.
Okay, bullet one. We're talking about these rare, rule-breaking immune rebels that lack standard uniforms, known as DNT cells.
Right.
Bullet two. They are split into two family trees, alpha beta and gamma delta. And bullet three, because science finally decided to look in the actual tissue instead of just the blood, we realize these rare cells are heavily concentrated right on the front lines.
Perfect summary.
Which leads to a question I want you, the listener, to ponder for a second. Have you ever considered that in any complex system, the rare outliers might actually hold the most power just because they operate out of sight? Think about how that applies beyond just biology.
It really is a profound shift in how we view complex systems. Because once you find them in the trenches, the next logical question is, well, what exactly are they doing there?
Right.
So the researchers took those 150,000 cells and mapped out 14 distinct functional subsets.
14.
Yeah. 6 from the alpha-beta family and 8 from the gamma-delta family. And here is where the divide between the two family trees becomes really stark.
Oh, because they have completely different day jobs.
Exactly.
Looking at the data, the alpha-beta cells seem to act mostly as regulators and managers.
Yes, their internal profiles show they are heavily involved in immune modulation.
Right, so they are keeping the environment stable, sort of regulating the other cells.
Yeah.
But the gamma-delta cells, they are the heavy-hitting assassins.
Highly cytotoxic.
Meaning their entire existence is geared toward actively hunting and killing target cells. So we've got managers and assassins.
It's a very apt description, but you know, cells don't just spawn into existence fully formed with a career path. They have a life cycle.
Right.
And to figure out how these cells grow up and choose their careers, the researchers used some incredibly advanced computational algorithms. Tools like Dynamo and Svelo that analyze something called RNA velocity.
Okay, RNA velocity. Let's break that down because the concept behind it is actually brilliant. It's essentially a biological time lapse, right?
Exactly. Think about how a cell operates. When a cell decides to turn on a specific gene, it first prints a raw, unedited copy of RNA.
Okay.
Then it splices and edits that raw copy into a mature form, which is eventually used to build proteins.
And because that editing process takes time, measuring the ratio of raw, unspliced RNA to mature, spliced RNA in a single cell acts as like a predictive compass.
You nailed it.
So it's like walking into a kitchen. If you see flour everywhere, water boiling, and dough rising on the counter, you don't need a time machine to know that bread is in the future.
Right.
You are seeing the raw materials mid-process. By looking at the raw RNA, these algorithms can predict exactly what a cell is about to become.
That is a perfect analogy. You are literally looking at the momentum of the cell. Yeah. And when the researchers applied this time-lapse algorithm to the alpha-beta managers, they saw a very straightforward path.
Oh, really?
Yeah. They start out naive in a subset called T2LEF1 plus CCR7 plus motto.
Wow, okay, T2LEF1 plus CCR7 plus. That is quite the biological barcode.
It really is. But basically from that naive start, they walk a highly predictable, straight trajectory toward a terminal, exhausted state.
Oh, man, so they do their managing, they slowly run out of steam, and they burn out.
Pretty much.
A very standard, almost depressing corporate life cycle. But the gamma-delta assassins, their trajectory is much more chaotic, isn't it?
Much wilder. The gamma-delta cells start out in an intermediate state, labeled T10GZMK plus way, where they are already armed.
Armed with what?
They contain chemical weapons called granzymes, which they use to force a target cell to self-destruct. So they begin their journey armed and dangerous, but then their developmental path hits a massive fork in the road.
It's like a biological choose-your-own-adventure book.
Yeah.
Starting at the exact same point, their internal choices change their ultimate destiny.
And those destinies are polar opposites. The first path trajectory 1 turns them into super killers.
Nice.
As the cells travel down this trajectory, they massively upregulate a specific master switch gene called TBX21.
Okay, TBX21.
Yeah. This switch locks them into becoming fierce, highly effective executioners that actively hunt the tumor.
That's the path we want them on. But the second path takes a very dark turn, right?
It does. Instead of the killer switch, they go down trajectory 2 and upregulate a different gene called NR4A1.
Which is an exhaustion switch.
Exactly. In the context of fighting cancer, it's absolutely the villain of the story. It acts as a burnout mechanism.
So it makes the cell profoundly exhausted and completely dysfunctional.
And worst of all, immunosuppressive. The cell doesn't just stop fighting the tumor, it actively starts suppressing the healthy immune system around it.
Wow. Giving the tumor a safe haven to grow.
Exactly.
Okay, so if you want a quick way to remember that fork in the road, think of the TBX switch as making them the best executioners.
Yeah.
The other path, the NR4A1 path, just leads to total burnout and betrayal.
That's a great mnemonic.
All right, so time for another quick 5-minute recap for you listening. Bullet 1, we have 13 different subsets of these D and T cells.
Yep.
Bullet 2, the alpha-beta ones manage the environment until they tire out, while the gamma-delta ones are armed assassins.
Right.
And bullet 3, those gamma-delta assassins face a critical fork in the road, choosing between fierce cytotoxicity on trajectory 1 or immunosuppressive burnout on trajectory 2. Which brings up my next question for the listener. If you were a scientist designing a cancer therapy, what mechanisms would you explore to barricade that second path and force all gamma-delta cells down trajectory 1?
Because that is exactly where the field of oncology is heading right now.
I bet.
But, to do that effectively, we really need to understand the absolute peak of what these cells are capable of. Let's zoom in on one specific subset from that gamma-delta family.
Okay, out of the 14 total subsets, there is one that just completely defies conventional immunology.
Yes.
It's the subset called T14 HLA-DR plus race C, and it acts as the ultimate multitasker.
The multitasker, exactly. To understand why this specific cell is so mind-bending, we need to talk about antigen-presenting cells, or APCs.
Okay, lay it on us.
Normally, when a tumor is silently growing in the body, the regular immune system doesn't automatically know what it looks like. It needs intelligence. So classic APCs like dendritic cells or macrophages will roam around, gobble up pieces of a dead tumor, and display those pieces on their surface using MHC class II molecules to warn the rest of the immune system.
Wait, this is where the conventional rules completely fall apart for me.
How so?
Well, classic APCs are like police sketch artists, right? They find a piece of the criminal, they draw the picture, and they hold it up to show the SWAT team, the regular CD4 and CD8 T cells. So the SWAT team knows who to attack.
Right.
The sketch artist doesn't kick down doors. But from what the data shows, are you saying this specific T14 gamma-delta cell is a sketch artist that is also a highly trained sniper?
That is exactly what makes it so revolutionary.
No way.
Using something called sum analysis, a type of machine learning to find hidden patterns in massive data sets, the researchers compared this multitasker to those classic sketch artists.
And what did they find?
Classic APCs digest the threat, display it, and their job is basically done. But this rare T14 cell uses a completely unique mechanism called endocytic cycling.
Okay, endocytic cycling. Let's dig into the mechanics of that because it sounds like cellular conveyor belt.
That's a brilliant way to visualize it. Endocytosis is the cellular equivalent of swallowing something whole. The cell binds to a piece of the tumor and engulfs it, pulling it inside its own body.
Right.
Cycling means it recycles it. Instead of just destroying that tumor fragment for food, it loads the fragment onto its display complex and physically pushes it back up to its outer surface membrane.
Mounting it like a warning flag.
Yes. It shows the antigen to CD4 and CD8 T cells to activate them. But because it is inherently a gamma-delta T cell at its core, it never loses its own direct cell-killing abilities.
That is insane.
Yeah.
It raises the alarm, recruits the massive regular army of T cells, and continues to actively shoot at the tumor simultaneously.
It completely eliminates the middleman.
The sketch artist with a sniper rifle. By combining the detection of the problem directly with the execution of the solution, the efficiency must skyrocket.
Oh, it does, and there is actual clinical data backing up how important this multitasker is for human patients.
Really, like real-world outcomes.
Absolutely. The researchers looked at clinical data from patients with bladder cancer and melanoma who were receiving modern checkpoint inhibitor immunotherapies.
Right.
They found that patients who naturally possessed high levels of this specific T14 multitasker subset in their tumors had vastly improved responses to the therapy.
Wow. Okay, let's synthesize that for our third recap. Bullet 1. T14 HLA-DR+ is a rare gamma-delta cell that breaks all the rules by multitasking.
Right.
Bullet 2. It acts as both an antigen presenter, using endocytic cycling,
Yep.
and a direct killer.
Yep.
And bullet 3. Having high levels of this specific multitasker is a massive predictor of success for patients on immunotherapy.
That's spot on.
It's wild to think about how much faster any system operates when the alarm system and the response team are exactly the same entity. Which leads to my question for the listener. Look at your own workflow. What happens when you combine the detection of a problem directly with the execution of the solution, just like the T14 cell?
It really is the pinnacle of biological efficiency. But, biology is highly dependent on environment. For our final exploration, we need to shift our geography entirely.
Where are we going?
We are traveling down to the gut to look at colorectal cancer. Here, we meet another specific subset, the T11ITGA1 plus cell.
Okay, T11ITGA1 plus.
Right. It's a gut-resident gamma-delta T cell. And what the researchers did here was physically map exactly where these cells live using something called spatial transcriptomics, specifically a platform called Visium HD.
Spatial transcriptomics. So instead of just grinding up a tissue sample and getting a raw list of what cells are present, this technology gives you a map. It's essentially like having Google Earth for the tumor microenvironment.
That's exactly it. Traditional sequencing loses the coordinates. You know the cells are there, but you don't know who their neighbors are.
Right.
By using spatial mapping on microscopic slides of the intestine, we can see exactly where every single cell is located. And if we connect this to the bigger picture, what they discovered is the ultimate biological Jekyll and Hyde story. Oh, I love a good Jekyll and Hyde analogy.
This single type of cell has two distinct faces, depending entirely on its zip code.
Location, location, location.
Exactly. The researchers found two functional states for this cell. Let's call them gray area M1 and gray M2. Gray M1 is the protector state.
Okay, gray M1 is the protector.
When the spatial mapping located these cells in healthy, normal intestinal tissue, they were in this gray M1 form. And in this state, they are actively producing anti-tumor cytokines, specifically ones called IFNG and TRAIL.
So they are maintaining order, keeping the tissue healthy, fighting off microscopic threats. Good guys.
Exactly. But when they map the exact same type of cell, literally right next door, inside the colorectal tumor.
Let me guess, it was a totally different story.
Inside the tumor, they transform into the gray M2 state, the traitor state.
Wow.
In this state, they flip their function entirely. They start producing pro-inflammatory, tumor-promoting cytokines, like IL-17A. Instead of fighting the cancer, they actually help the tumor grow. Okay, we really need to pause and explore the how here. How does a fundamentally good cell go so completely bad? I mean, how does a tumor manage to brainwash a highly trained protector into becoming a traitor?
It comes down to the harsh reality of the tumor microenvironment and cellular metabolism.
Okay.
Solid tumors are intensely chaotic. They mutate and grow so fast that they vastly outstrip their own blood supply. Because the blood vessels can't keep up, the environment deep inside the tumor becomes highly acidic and severely hypoxic.
Hypoxic meaning it lacks oxygen.
Exactly. It is starved of oxygen.
So the cell enters a literally toxic work environment.
A toxic and suffocating environment.
Yep.
When the healthy GURM1 protector cell wanders into this hypoxic tumor core, the lack of oxygen triggers a massive internal panic response, specifically a hypoxia signaling pathway driven by HIF1A activity.
Okay, HIF1A panics the cell.
Right. And without oxygen, the cell can't burn fuel efficiently in its mitochondria anymore. To survive, it is forced to radically change its diet. It switches its entire metabolism to a process called glycolysis, which is basically fermenting sugar in the cytoplasm for quick, dirty energy.
It's like a person pulling an all-nighter in a high-stress environment, panicking, and eating nothing but pure sugar and junk food.
Yes.
Eventually, that toxic diet completely alters your mood and behavior.
That is exactly what happens at a genetic level. This forced metabolic switch to dirty energy alters which genes are turned on and off. The metabolic reprogramming literally rewires the cell's identity, brainwashing the GURM1 protector into the GURM2 traitor.
That is staggering. The environment forces a change in diet, and the diet changes the core identity of the cell.
It really is. And they proved this wasn't just some theoretical mechanism happening in a petri dish. They tested this in vivo in a living mouse model with MC38 colon cancer.
Oh, it happened.
To prove just how vital these specific gamma-delta T cells are, they took these mice and treated them with standard anti-PD-1 immunotherapy. Normally, with the immune system intact, the drugs worked and the tumor shrank beautifully. But in a separate group, they systematically depleted just these rare gamma-delta T cells first, and then gave the exact same immunotherapy.
And without them, the immunotherapy totally failed, didn't
It
it?
completely failed. Without these rare 1 to 5% outlier cells, the cutting-edge cancer drugs were effectively useless.
Okay, time for our final recap. Bullet 1. T11 ITGA1 plus is a gut-resident cell that exists as the protective GRM1 in healthy tissue.
Yep.
Bullet 2. Tumor hypoxia and acidity force the cell into glycolysis, brainwashing it into the traitorous GRM2.
Right.
And bullet 3. Removing these shape-shifting cells completely breaks modern immunotherapy in mice.
Exactly.
Which raises a truly philosophical question for you listening.
Right.
How much of this cell's identity is its internal programming versus the environment it lives in? It's the ultimate nature versus nurture debate playing out on a microscopic battlefield.
It really is.
Okay, we've covered incredible ground today. We started with the basics of these rule-breaking DNT cells. We explored their distinct trajectories as managers or assassins. We unpacked the multitasking sketch artist with a sniper rifle. And we just revealed the shape-shifting traitor of the gut.
We did.
So, pulling back from the single-cell level, what does this all mean? When you zoom all the way out to the pan-cancer clinical data, what is the ultimate takeaway for human patients today?
Well, the researchers synthesized all this massive data by grouping human patients based on the specific mixture of these cells inside their tumors.
Okay.
They identified one specific group where patients had a steady, highly active infiltration of those anti-tumor gamma delta cells, the snipers, the protectors. They classified this as the Ottkunstflux group.
Quite a name, but the result.
Across the board, whether you are looking at kidney cancer, skin cancer, or stomach cancer, the patients in this specific group have significantly better overall survival rates.
Wow. It proves that the presence of these rare wildcard cells is a massive independent predictor of surviving cancer.
Yes. They aren't just a biological curiosity anymore. They are a critical foundational lever in the human immune response.
Right.
If we can design next-generation immunotherapies that specifically target and boost these DNT cells, maybe a drug that forces them down the superkiller TBX21 path, or a drug that prevents the metabolic brainwashing in the tumor core, we could unlock a completely new frontier in oncology.
We started today's deep dive by talking about the immune system as an organized military and how the DNT cells were the rebels without a uniform. But what we've discovered is that these rebels aren't just causing chaos, they are running the most sophisticated, high-stakes special ops missions in the entire human body.
They really are the special forces.
As we wrap up, I want to leave you, the listener, with one final thought to mull over. We just learned that the physical microenvironment of a tumor, specifically a lack of oxygen and high acidity, can literally hack an immune cell's metabolism and turn a fierce protector into a tumor-promoting traitor. It's an invisible war decided almost entirely by local chemistry. If metabolism and local environment can completely rewrite the immune system at a cellular level, how much does our macroscopic environment, our daily metabolic choices, the air we breathe, the ambient stress we endure, quietly dictate the unseen wars happening inside us right now? Until next time, keep diving deep.